Last time we did the Gram-Stain, and if you remember, the Gram-Stain classified bacteria into gram-positive or gram-negative. Now the Gram-Stain only goes so far as to classifying a bacteria. It's great as a first step, and remember, it's really important in knowing whether a cell is gram-positive or gram-negative to determine the antibiotic use. However, there are tons of bacteria that are gram-negative, tons that are gram-positive, tons that are staphylococcus, and so on and so on. So just because you have a gram-positive staphylococcus doesn't mean you know exactly what it is. Or you have a gram-negative raw bacillus, you still don't know what it is. Okay? So one of the next diagnostic tools that can be used is called an acid-fast stain, and that's what we're doing today. The acid-fast stain is used for a specific group of bacteria. The group of bacteria that the acid-fast is used for is the genus Mycobacterium. In the Mycobacterium genus, there are two major bacteria that can cause disease. The first one is Mycobacterium tuberculosis. You can imagine that causes tuberculosis. The other one is Mycobacterium leprae, causes leprosy. These bacteria are very specific, are special in the fact that they have an outer waxy coat outside their wall called mycolic acid. Mycolic acid is highly resistant, and you just can't give it any antibiotic to kill it. So if a person has either tuberculosis or leprosy or one of those types of diseases, you can't treat them with a regular antibiotic. You have to figure out an antibiotic that either breaks down that mycolic acid or penetrates to get to the wall, okay, the regular wall. So we're going to do an acid-fast stain. Now, just like it sounds, acid-fast bacteria will retain their wall components, therefore retain the primary stain even when washed with acid. Okay? Other bacteria that don't have that mycolic acid, if you were to put the acid alcohol on them, it's going to wash off. Okay? Now for the acid-fast stain, we're going to do something a little bit different than we did with the Gram-Stain. We're going to use heat. So for this, one of the things you want to think about is I already have this on. It's been on for a while. We all know how long water takes to boil. So you don't want to wait till the last minute to start boiling your water. Okay, you want to have it boiling. Now, you don't actually see it boiling, but you can see steam is coming off of it. So we want it just enough. It doesn't have to be any kind of rapid boil. If you get it boiling too much, you want to turn it down. The idea is you just want steam coming off of it. And so what we're going to do here is we're actually going to steam our bacteria. I'm going to move this a little bit closer. All right, and that's where we start sweating. So I have my bacteria, my slides. Now remember, with all procedures, all staining procedures, you're going to fix them before you, I'm steaming up from the steam, you're going to fix them before you ever go to stain them. The only time you're not going to fix them is if the procedure says do not fix, okay, which we'll see later. So what we're going to do is I have my bacteria, I have it prepared, I've made the smear and I've fixed it. My left goggle is really steaming up. So now I'm going to gently place the bacteria, or the slide with the bacteria fixed on it. It's already fixed. I've already done the Bunsen burner. I've fixed it. Now you have condensation forming underneath there, and I can see the steam coming, so that's hot enough. The reason that we're going to use steam is because that mycolic acid is so resistant and so thick, it's a waxy coat, that we want to kind of soften it a little bit to allow the stain to penetrate. So the method that we're going to use requires the steaming. And you'll notice I have the beaker with the water there already. You want to put the smears that are fixed on the beaker before you add any stain. Okay? Now when you go to add the stain, the primary stain of the acid-fast stain is the carbonyl fusion. Okay? We saw this when we did simple staining. Okay? So what we're going to do is we're going to add the carbonyl fusion. Add just enough to cover where you think the smear is, where your circles are, but try not to drop too much of it in the water. Now that's going to steam for 5 minutes. I'm not going to sit here for 5 minutes steaming, but one of the things I want to point out is as you're doing this, you want to make sure that the carbonyl fusion doesn't dry out, and steam is going to make it dry out pretty quickly. So while it's steaming for the 5 minutes, if you notice it starts to dry out, add some more. Okay? But once again, we don't want too much in the water. If you get a little bit, that's okay. But if you have a big red beaker when you're done, it's too much. The size of beaker that I'm using is a 400 ml beaker. It's the perfect size for allowing you to put the slides on top. So that's going to steam for 5 minutes. I'm just going to let it steam for a little bit and talk about the rest of the reagents. The next reagent we're going to use for decolorizing is going to be the acid alcohol. If you remember with the gram stain, we used ethyl alcohol, okay? This time we're using acid alcohol because we want to see if the bacteria retain their primary color when washed with acid. The counterstain in this case is going to be methylene blue. So if a bacteria is acid-fast positive, it will be red at the end of the acid-fast stain. If it is non-acid-fast or negative, it will be blue. Now remember with the gram stain, purple was positive, red or pink was negative. So you need to be thinking independently when you talk about a stain. Some people just automatically red is negative, okay, and they put the gram stain and acid-fast stain together. They're completely independent of one another. So red in this case actually means positive, okay? So that has been steaming for 5 minutes. So what we want to do is gently remove the slides. Now this is hot, but it's not so hot that I can't touch the edges of it. You want to be careful not to get too much of the dye in the water. You notice I didn't burn myself. It's not extremely hot because I don't have this rapid boil going, okay? So don't be too afraid of it you're probably not going to burn yourself. If you fear that you might burn yourself, you could use clothespins, but they're real cumbersome, and the majority of the students, when they go to use the clothespins, end up knocking it into the water. Then they have to start over. So if you're real gentle with taking it off, you don't have to worry about that. So that has steamed for 5 minutes. Now I'm going to rinse, just like we do. We always rinse with water between every step. So I'm going to rinse that off. And just keep rinsing. Don't be afraid to use too much water. It's okay, right? Now with the procedure, you have a couple of bacteria that you're going to use. It's up to you how you want to do it. You can put both of them on the same slide, or you can put them on separate slides. Remember, it's important to label properly. When you go to label your slides, you can abbreviate it like MS for Mycobacterium smegmitis, okay, which is the bacteria we're going to use. Not all Mycobacterium are pathogenic. We are using a non-pathogenic Mycobacterium called smegmitis. When you label them, make sure once again that you label on the back and you label clearly and you know what you're writing down. The stock tube is labeled and the writing on it is sometimes interpreted differently. Make sure you have the correct bacteria and that you label it correctly. You don't write a letter of a word that's not a bacteria that you're using. So make sure you know ahead of time what bacteria you're using and the bacteria that you're taking off the cart is the correct bacteria. Now our decolorizing step is similar to what it was in the gram stain. We're going to squeeze the alcohol on there, the acid alcohol, just like we did before, until it almost runs clear. Some of us, like myself, I believe I've washed the color off of the back. You want to do that until it begins to run clear. Many of us realized with our gram stain that we had decolorized too long because our gram-positive cells were pink. So the same thing with this, you don't want to decolorize too long. I want to point out when you're using the acid that it has a tendency to wash a lot of things off. So make sure you don't get all of, when you label, maybe label at the top so that you're not getting acid on it, even though it's on the back. Then we want to immediately wash with water, okay, to stop the decolorizing. Ooh, that smells lovely. All right, the last thing that we're going to do is the counterstain. In this case, it's methylene blue. So you put that counterstain on for about a minute, 30 to 60 seconds, depending on what your lab procedure says. It just depends on the lab manual which procedure you're going to use. So you let that sit for its time frame. Then you're going to rinse it like we've done in the past. Once again, don't be afraid to rinse it all off. When you've seen slides that have too much stains left on them, you end up with big blotches of stain. Remember, that's not bacteria, okay? So we've rinsed them, now we can blot them dry as we did in other procedures, okay? So remember, you want to gently blot with a paper towel, Don't rub, because you don't want to rub off the smear that you just spent 45 minutes on, or however long it takes you. Okay? We'll let those air dry completely before we look at them on the under the microscope.